Okay, so what I have here, I'm going to talk about osmosis. And I have 3 different cells. And each one of these cells are, have like the simplest ingredients listed. Here I have, this cell has 0.9% salt and 99.1% water. We're going to ignore all the other ingredients right now. But look at that, adds up to 100%. Okay, and actually this is the normal amount of salt in a normal cell, 0.9% salt. So if we take that cell and we put it in a solution that has 0.9% salt and 99.1% water, we can see that the concentration is the same on the inside and the outside. Osmosis says water moves from high concentration to low. So here I have water moving, 99% water, 99% water. Every molecule of water that moves out of this cell, it's going to cause another molecule to move in order to keep the concentration the same on both sides. We call this kind of solution that's the same on the inside and the outside, we say we've put it in an isotonic solution. And for example, in clinical settings, you might use something called normal saline to dilute medications or to rehydrate patients. You want to put the same amount of salt in their body. So if their water concentration in their cells are low because of dehydration, then you put them in 0.9% salt, normal saline, and they'll rehydrate. If we look over here, here's our same cell, 0.9% salt, 99.1% water. Now if we take that cell and we put it in 100% water and 0% salt, osmosis says water moves from high concentration to low. So here I have the 100% water is going to move into the cell. Now remember that human cells do not have cell walls. They don't have barriers on the outside. They do have a plasma membrane or a cell membrane, but they don't have a box on the outside like a cell wall. So water is going to move into the cell. It's going to move from high concentration, 100%, to lower concentration, 99%. It's going to cause the cell to swell up and potentially burst if enough water moves into there. With human cells, like our own cells, we have to keep our cells in an isotonic environment, but or else they're going to burst. If you're a plant, you have a box around your cells. You have a cell wall, and therefore the cells will not burst in a hypotonic solution. They kind of look pretty much the same. In our last cell, again, it's the same cell, 0.9% salt, 99.1% water, but this time we put the cell in a hypertonic solution. And hyper means high salt, so if I have 3% salt and 97% water, 100% total, osmosis says water moves from high concentration to low. Here's my high concentration of water. Here's my lower concentration. Water's going to move out of this cell, and I'm going to move water out of the cell. It's going to shrivel it up. And hypertonic solutions can be deadly. They're not something that's desirable for a cell. You don't have enough water, it can't conduct its metabolism. So the 3 conditions, if you look at those again, osmosis, isotonic cell, the water's going to move in and out at equal amounts. The cell's going to look normal. A hypotonic, a low amount of salt, high amount of water. Water's going to move into the cell. The cell's going to swell. If it is a plant cell, it'll look pretty much the same because it has a cell wall. If it's an animal cell, it's going to pop. And hypertonic, high amount of salt outside, so low amount of water. So water's going to move out of the cell. Hope that helps.